They are injecting salmon DNA into human faces. Not a metaphor, not a supplement, not a derivative or an extract. Actual polynucleotide chains, long sequences of nucleotides harvested from the genetic material of Pacific salmon, drawn into a syringe and delivered into the dermis of a human face. And it works. Not because the salmon DNA replaces anything in the human tissue, not because it integrates, not because it becomes part of the person receiving it. It works because of what the human body does when it finds it there. What the body does is the story.
A polynucleotide is a chain of nucleotides. Each nucleotide is a three-part structure: a phosphate group, a sugar, and a nitrogenous base. String hundreds of them together and you have a polynucleotide. String billions of them together in the right sequence and you have DNA. The polynucleotides used in this treatment are not a complete genome. They are fragments, long chains of nucleotides extracted from salmon milt, the seminal fluid of male Pacific salmon. Salmon milt is used for one reason. It is one of the most abundant natural sources of highly polymerized DNA available at scale. The extracted fragments are purified, sterilized, and processed into a gel or solution. What enters the syringe is not alive. It carries no salmon genetic instructions. It will express no salmon proteins. It is raw molecular material. And the human dermis has no idea where it came from.
The needle enters the dermis, the layer of skin beneath the epidermis, rich in collagen, elastin, fibroblasts, and blood vessels. The polynucleotide solution is deposited in small volumes throughout the target tissue. In the seconds after injection, the surrounding tissue encounters something it was not expecting: long chains of extracellular DNA, fragments of nucleic acid present in the intercellular space where they do not normally belong. And the tissue responds the way it has been responding to extracellular DNA for 500 million years, as though something has been damaged. Because in biological history, extracellular DNA has meant one thing: cell death, tissue injury, a wound that needs repairing. The presence of DNA outside cells is a damage signal, an alarm. The tissue does not interrogate the origin of the signal. It responds to the signal itself. Fibroblasts, the connective tissue cells responsible for building and maintaining the extracellular matrix, begin to activate.
The specific mechanism involves a receptor called the A2A adenosine receptor. When polynucleotide chains are degraded by extracellular nucleases, enzymes that break down extracellular nucleic acids, the degradation products include adenosine. Adenosine is a purine nucleoside. It is also a signaling molecule with well-characterized receptor targets throughout the body. The A2A receptor, when activated by adenosine, triggers an intracellular signaling cascade involving cyclic AMP, a second messenger that activates protein kinase A. Protein kinase A phosphorylates transcription factors, specifically CREB, that enter the nucleus and upregulate the expression of genes encoding growth factors. Among the most significant, vascular endothelial growth factor, VEGF, which stimulates new blood vessel formation in the treated tissue, and fibroblast growth factor, FGF, which directly stimulates fibroblast proliferation and collagen synthesis. The polynucleotide chain did not build new collagen. The adenosine signal did not build new collagen. The growth factors built the collagen, and the growth factors were produced by the human fibroblast in response to a receptor signal triggered by the breakdown products of salmon DNA. 4 steps removed from the injection, entirely biological, entirely the body's own work.
The fibroblast that received the growth factor signal begins producing procollagen. Procollagen is the precursor to mature collagen, assembled inside the fibroblast, packaged into secretory vesicles, and released into the extracellular space. Outside the cell, procollagen is cleaved by specific metalloproteinases, the propeptides removed, releasing tropocollagen.
tropocollagen molecules self-assemble into collagen fibrils, a triple helix structure, 3 polypeptide chains wound around each other, approximately 300 nanometers in length and 1.5 nanometers in diameter. Fibrils aggregate laterally into collagen fibers, the structural architecture of the dermis. This new collagen occupies the extracellular matrix, where older, degraded collagen fibers had left gaps, the structural deficit that manifests on the surface as the visible changes of aging skin. The salmon DNA is long gone, degraded, its nucleotides reused. What remains is new collagen built by human fibroblasts responding to a biological signal that the salmon DNA set in motion.
The body does not know it has been prompted. It knows only that extracellular DNA was present, that the A2A receptor was activated, that growth factors were expressed, that fibroblasts were signaled, that collagen was needed. It responded to each step in sequence, the same sequence it has run every time a wound has needed repair since long before the species using it had a name for any of the molecules involved. The aesthetic medicine industry calls this biostimulation, stimulating the body's own repair mechanisms rather than replacing what is lost. The biology calls it what it has always been, a repair signal, received and answered. The salmon had nothing to do with it. The body had everything to do with it. It always does.